Hello everyone, and thank you all for joining us for our weekly Hanauma Talk seminar series presented by the Hawaii Sea Grants Hanauma Bay Education Program. My name is Gavin Y, and I am the Outreach Program Coordinator for the Education Program. First off, I'd like to introduce Maya Walton, who is the Hawaii Sea Grant Program Leader and Research Coordinator. She acts as a liaison between various researchers, Sea Grant Extension faculty, and University of Hawaii Sea Grant Program. She also serves as a programmatic point of contact for several fellowships managed by Hawaii Sea Grant, such as the cadre of Sea Grant graduate fellows we are featuring tonight. A few reminders before Maya introduces our speakers. Please remember to keep your questions till the end of the presentation, or please type them in the chat room at any time. Also, please remember to turn off your microphone and video during the presentations. We appreciate it. With that, Maya, would you please talk about a little bit about the Sea Grant Graduate Fellowship Program, as well as introducing our speakers for today?
Thanks, Gavin. Yeah, happy to talk about our Sea Grant Graduate Fellows. And we also have PICAST graduate scholars tonight who are sharing their research with us. So aloha, my name is Maya Walton. I work at the University of Hawaii Sea Grant College program as the program leader and the research coordinator. And I see many familiar faces. I know some of you have seen these Thursday night talks. We've been featuring the Sea Grant Graduate Fellows and the PICAST Graduate Scholars during the months of November and December. And tonight, it's kind of bittersweet actually, is our last set of 3 talks from our fellows, from our graduate students. So we're really excited to hear this last set of 3 tonight. Okay, so a little bit more about these Sea Grant Graduate Fellows. They're engaging in really exciting research at but they're also receiving training in science communication, and we asked them to complete 50 hours of community outreach a year, and we asked them to get their science out into the general public. And one of the ways they do this is through talks just like this one, outreach talks just like this one, through Hanauma Bay's seminar series, the lecture series. So we have a really great set of talks tonight, and I'm really excited to introduce, again, our Sea Grant Graduate Fellows and our PICAST Graduate Scholars. PICAST, by the way, I forgot to mention, that stands for the Pacific Islands Climate Adaptation Science Center. So I'm excited to have Sea Grant and PICAST represented tonight. First, we're going to hear from Sea Grant Graduate Fellows Emily Conklin and Cassie Kaʻapu Lyons. Their talk is titled Setting the Table for Aquaculture. What do baby fish eat? I hope there's lots of baby fish photos. Super excited to see that one. Our second talk tonight is going to be from Sea Grant Graduate Fellow Tineill Dudoit. And her talk is titled Exploration of Geoscience Through Place and Culture. And our last speaker this evening will be Sea Grant Graduate Fellow Gina McGuire, and her talk is titled Modeling the Benefits of Agroforest Reforestation in Hawaii, a Land to Sea Approach. So like Gavin said, we're going to take questions in the chat as we go along, so feel free to type those in. And we'll take one question as we move between speakers, but the bulk of the Q&A will be towards the end of our 60 minutes together tonight. Okay, so when Emily and Cassie are ready, the floor is yours.
Okay. Of course. All right.
We saw the slides earlier, so we know the first slide is really, really pretty. Looks good.
Let's see. All right.
Okay, looks good, Cassie.
All right, you ready? Hey everybody, my name is Emily Conklin, and I am a PhD student at the Hawaii Institute of Marine Biology.
And I'm Cassie, and I'm also a PhD student at the Hawaii Institute of Marine Biology. And tonight we're going to share a little bit about our work with baby fishes. So this is probably what people are most familiar with when they think of fish. We see them in our markets and on the dinner tables and in our aquariums. But unlike freshwater fishes, marine fishes are largely collected from the wild. And so here's a simple figure kind of showing the increase of demand with the decrease in supply.
So to keep up with this increasing demand, many have started to turn to aquaculture as a valuable alternative to wild-caught fish. And aquaculture has the potential to reduce fishing pressure on some of our dwindling fish stocks.
It also can help supply food for growing human populations that are heavily reliant on fish for food sources and create job opportunities for coastal economies, both here in Hawaii and across the nation.
So one thing that makes marine aquaculture tricky is that many marine fish have really complicated life cycles. So right here, we're looking at a manini. They start off as tiny little larvae, and they go through a few stages from larvae to juvenile before finally growing up to adults. And all of these stages can have very different lifestyles. So they might live different places, eat different things, have different behavior. And that makes aquaculture tricky because we have to figure out how to properly care for them at every stage.
Although the ability to breed fish in captivity has increased over the last few years, very few captive raised marine species are available commercially. And this high mortality is due to disease and starvation. Disease can be controlled with sanitation and remedial measures. However, starvation remains difficult to control, largely due to our lack of knowledge about what baby fish eat. And because of this, larval nutrition and feed formulation play an essential role in the aquaculture industry. So for this study, we focused on what do larval fish eat in the wild.
So for adult fishes, there are a few standard methods for figuring out what they eat. So one is actually getting in the water and observing them in the wild. Or we can conduct feeding trials where we basically give them one type of food after another and see what they like to eat. And another, I know this picture is a little gross to look at, but is a method of actually dissecting the fish and getting a look at what's inside the stomach to figure out what they're eating in the wild.
Methods used for analyzing stomach contents in adults have its drawbacks. However, observing larval fish foraging in their natural environment is nearly impossible due to their small size. And feeding trials are time-consuming and tend to be very simplistic in design. The stomachs of larval fish are tiny, and their contents usually consist of soft-bodied microscopic zooplankton in various stages of decomposition, which results in this unidentifiable muck. Prey that are complete enough to identify require taxonomic training.
So because of the problems with applying these standard methods to baby fish, our research is trying out a new approach and figuring out what they eat. So there have been a lot of advances in molecular technology recently that offer promise for fast and cost-effective diet analysis.
And these advances give us the potential to circumvent many of the challenges presented by the traditional diet analysis. We have the potential to take small amounts of DNA that are left in the gut and examine the presence and absence of a full range of prey items. without cutting open individual stomachs. It also allows us to detect majority of the unseen prey items that are digested and otherwise unidentifiable, and allow for examination of multiple species and developmental stages in a single shot.
So we can think of DNA a little like a barcode at a grocery store. So DNA sequences are unique to a particular organism, just like barcodes are unique to a certain product. If you were to go to the store and cut all of the barcodes off the products, you can still tell what products they came from.
And we can take these short DNA sequences and link them to the organisms that they come from, matching the otherwise mystery DNA found in unidentifiable contents of a stomach to, say, the krill that organism has eaten.
So in order to study what the larvae are actually eating, we first have to go get some. So the first step in this process is going out and collecting larvae from the wild. In this case, we're catching some baby manini. So once we have these baby fish, we take them back to the lab. They're sometimes not much bigger than your fingernail, so we can't actually dissect them like we would with adult fish. So we homogenize them instead. And homogenizing is just a fancy word for basically blending them. And once we have our homogenized fish, we can then extract DNA from them.
The extracted DNA is then put into a PCR machine to make many copies of a target DNA sequence for analysis. And then the sequences are put into the sequence that reads the DNA and puts it into a language we can interpret. The DNA then is returned to us and separated into unique barcodes.
Since we have our unique barcodes, we can take them and match them to each potential food species that the baby fish might be eating.
A barcode database is kind of like a molecular dictionary. It's where each molecular sequence can be matched up to its organism. And several databases already exist and are made available for use, including the Barcode of Life and NCBI's GenBank.
So our main goal right now is to see if this method actually works. So what we have now is super preliminary data just from a small sample of baby fish. Basically a proof of concept to see if we can actually get some results out of it. And the first question we have is whether we can actually detect DNA at all from the prey. So this on the right is a bar graph showing the number of prey species identified per baby fish species.
And we were successfully able to detect DNA that was unique from the fish in a variety of fish species. For example, if we go back to the manini that we caught earlier, we were able to detect 5 unique DNA barcodes that were different from the fish. So will we be able to detect DNA of prey using this method? Yes.
So now that we know we have our prey DNA, our next question is whether we can use those DNA barcodes, take them back to the databases, and identify the actual food species. So this is a figure showing baby fish species on the left and connecting them with the prey species they're eating on the right.
And this plot, although it feels a little bit busy, allows us to visualize the links between different fishes and their diet. So to make it a little bit easier, we'll focus only on the one fish, our manini. And we can trace those 5 unique DNA sequences to a list of identified prey items on the right. Alternatively, we can look at how diets may be similar or different between fish species. So for example, in this preliminary set of data, 9 out of the 12 fishes had DNA of this species of krill, in their stomachs. So yes, we think we are able to examine the diet composition of fish larvae. Our goal of this project is to harness this advanced genetic technology for the benefit of aquaculture. Once we're able to identify what larval fish eat in the wild, we can set the table for aquaculture and work with growers to identify nutritional requirements in lucrative species that have been otherwise difficult to grow out in captivity. This will allow the aquaculture industry to greatly diversify.
So now that we have our proof of concept, we can start looking at our next steps. So one is talking to industry partners about possible species to focus on, as well as what their needs are. So we want to focus on species of cultural and commercial interest. So for example, popular aquarium fish like butterfly fish and yellow tang, as well as important food species.
We will use this molecular approach to further evaluate similarities and difference in larval fish diets between different fish species, and examine how diet changes through development in order to better close the life cycle in captivity. And with that, I'd like to thank our collaborators, our industry partners, the many students, labs, and staff that have helped collect samples and sort, and my funding sources, the National Science Foundation, Center for Tropical and Subtropical Aquaculture, and Sea Grant. And thanks to Frank Bonch for all the beautiful larval fish photos in this talk. Thank you for listening.
Thank you. Applause for Emily and Cassie. If you have a question for our first two speakers, please do put it into the chat. And while those questions come in, let's have our next speaker, Tineill, bring up her slides. Okay, well, I'll ask a question as we move to the next speaker. You guys talked about, part of this is collecting the wild specimens, collecting the larval fish. What is that like? When do you go? Where do you go? How do you know where and when to go to get these teeny tiny fish?
Most of the fish that I'm using for this study actually came from Kaneohe Bay, and I was going out 3 times a day bi-monthly for almost 2 years to collect fish larvae.
Wow, so this was a all the time effort for a whole year.
Yes.
Awesome. Okay, so that's what it takes to do this kind of research, is got to be out there collecting teeny tiny fish for many, many hours at a time. Okay, so Tineill, your slides are up, looks good, and whenever you're ready, you can go ahead.
Sorry, I'm trying to get.
No worries, we could hear you for a second.
Hello, can you hear me?
Yep, we can hear you and we see your slides as well.
Okay. Aloha aya'i kako. Good evening, everyone. My name is Tineill Dudoit, and I'm a graduate student at the University of Hawaii at Manoa. My presentation today is about the power of a place, teaching geosciences through place-based education. The overarching goal of my project is to promote place-based educational resources and practices in the School of Ocean, Earth Science, and Technology, or SOEST. By incorporating place-based education, or PBE, into SOEST courses, I hope to contribute to existing efforts within the school that are aimed at increasing recruitment of new SOEST majors from diverse backgrounds, increasing the retention of existing SOEST majors, and increasing the interest and engagement of all students enrolled in SOEST courses, especially those who have an Indigenous worldview. The anticipated long-term impact of my project will be to help prepare local, diverse geoscience workforce to help meet Hawaii's current and future professional needs. In this presentation, I define the word geoscience in the broadest possible sense to include ocean, earth, environmental, atmospheric, and planetary geosciences. The University of Hawaiʻi is the state of Hawaii's largest and only provider of public education, or public higher education. Established in 1907, the UH system has 10 campuses throughout the Hawaiian Islands, including 3 universities and 7 community colleges. Committed to making attendance affordable and accessible, UH has one of the lowest student debt ratios in the country. The flagship campus of the UH system, Manoa, is a land, sea, and space grant institution. Recognized as a doctorate graduating institution with very high research activity, it provides various opportunities for study and exploration. Manoa students come from within and beyond Hawaii, generating a large and diverse campus enrollment. UH Manoa is home to the School of Ocean, Earth Science, and Technology. Established in 1988, SOEST is a world-class research institution, research and academic institution with an annual budget of over $100 million in extramural research funding. It allows students and faculty to conduct rigorous scientific investigation on various topics. SOEST offers 12 degrees through 4 academic departments: atmospheric sciences, earth sciences, ocean and resource engineering, and oceanography. However, despite having the world-class status as a research institution, SOEST has a small student enrollment. Now, this is a chart for student enrollment of majors of UH Manoa, and listed on the left are schools and colleges within Manoa. SOEST is highlighted with the red box, and as you can see from this chart, SOEST has a small student enrollment. In fact, only 271 students, which includes 125 undergraduates and 146 graduate students, are enrolled in SOEST majors. This makes up only 2% of UH Manoa's total student enrollment. In addition, SOEST undergraduate enrollment is particularly low. Although UH Manoa has a high percentage, 72, of undergraduates, SOEST undergraduates make up only 46% of that school's total enrollment. Not only does SOEST have a low student enrollment, but it's not as diverse as UH Manoa. These are pie charts based on student enrollment at UH Manoa and SOEST. As you can see, there is a noticeable change in the percentage of Caucasians, Asians, and Hawaiians between these 2 pie charts. In particular, there are very few Native Hawaiian students enrolled in SOEST degree programs. Unlike Manoa, which has 15% Native Hawaiian student enrollment, only 6% of SOEST students are Native Hawaiian. This suggests that more can be done to increase Native Hawaiian enrollment and diversify, perhaps through recruitment and/or retention. Finding a way to prioritize engagement, personal learning experiences, and critical thinking that is meaningful to students could boost motivation and persistence in the study of geosciences. Place-based education, or PBE, is a teaching approach that promotes a connection to place through the incorporation of local knowledge and resources and serves as a foundation for curriculum development. Studies have shown that PBE increases interest and participation, improves student achievement, and increases recruitment and retention. Studies of PBE specific to geoscience report improvement in the understanding of scientific concepts, an increase in scientific self-confidence, and improvement in the desire to study geosciences. Examples of PBE in geoscience literature include curricula based on natural processes and cultural themes, sampling and monitoring programs, ecosystem restoration, and geotechnical application. PBE is an effective teaching method in geoscience. However, to gauge its significance in SOEST, an inquiry was necessary. starting with questions like, do students and faculty value PBE? How prevalent is PBE in SOEST? And does PBE improve students' learning experience? To answer these questions, Battier, Wilson, and Bruno conducted a survey of PBE in SOEST. In spring 2018, 2 online surveys, one for faculty and one for students, were designed to gain information relating to PBE and SOEST courses. The various question types that were utilized for each survey included Likert scale, open-ended, and multiple choice questions. The student survey included 14 questions, and the faculty survey included 13 questions. Courses that respondents reported on range from entry-level to graduate-level courses and included both lecture and lab styles. Surveys were emailed to SOEST faculty and students at the beginning of the fall 2018 semester, and data were collected and analyzed using SurveyMonkey software. Now let me direct your attention to the pyramid, or the figure of the pyramid on your left. This pyramid, created by Battier, Wilson, and Bruno, is a 3-layered pyramid framework for introducing PBE practices into the classroom. The pyramid is separated into 3 layers, starting from layer 1 on the bottom to layer 3 on the top. As these layers increase, from 1 being the lowest to 3 being the highest, so does the level of cultural competency. The first layer includes data sets, field trips, and guest speakers. The second layer includes field explorations, community workdays, and service learning. And the third layer includes indigenous knowledge, traditional practices, and Hawaiian terms. A total of 81 students and 59 professors' survey responses were analyzed. These results indicate that the survey respondents felt that PBE is important. Most participants found PBE to be an effective teaching and learning method. Positive impacts reported by students included the enhancement of student learning and increased interest in geoscience content. Almost all faculty respondents reported that they do incorporate PBE, and the most commonly utilized PBE strategies were data sets, guest speakers, and geoscience-related field trips. These PBE activities fall into the layer 1 category on the figure to the left. These results suggest that SOEST faculty lack confidence in using PBE practices that require a higher levels of cultural competency. Overall, the study indicates that more can be done to address the lack of resources and faculty confidence in generating culturally relevant teaching materials. So far, I presented background information and highlighted a recent study on PBE in SOEST. Next, I would like to focus your attention on the components of my research project. First, I compiled a resource repository of existing PBE resources currently being used in SOEST. Second, I expanded this repository to include new PBE resources. And third, I created Environmental Systems Research Institute, or ESRI, story maps to showcase some of the existing and newly added resource material. First, I met with 16 SOEST faculty within the 4 academic departments and asked them to share their PBE resources that they were currently using in their classes. A snapshot of the Excel sheet I created is shown in the slide. Resources included field guides, PowerPoint presentations, literature including lessons, books, journal articles, data sets and models that are rooted in place and/or Hawaiian culture. With their permission, I created an online repository of these resources and summarized them in an Excel sheet. Second, I expanded this repository to include additional place-based and/or culturally relevant resources that are not currently being used in SOEST, but are relevant to geoscience. The table displayed is a preview of the new resources that I added. Third, I created 6 ESRI story maps to showcase some of the place-based resources existing and newly added on the Google Drive. The table on the slide summarizes the title, the number of pages, and resources within each story map. The first 2 story maps, Exploring Hawaiʻi and the Pacific Through Hawaiian Language, Legends, and Belief Systems, and Exploring Hawaii and the Pacific Through through traditions, land use practices, and geoscience highlights selected resources such as books, journal articles, and newspaper articles. These two story maps are categorized as resource-specific story maps. Below is an example of what a page from these story maps looks like. This is only a glimpse of the information contained within a page. And on average, each page contains one full page or more of written information and resources. They are intended to give SOEST faculty a small taste of the many resources available on the Google Drive and to encourage faculty to explore this repository more fully. Each page in these two story maps corresponds to an individual resource on the Google Drive. The next three story maps are organized by geography and correspond to the islands of Maui, Oahu, and Hawaii Island. Below is an example of what a page from these story maps looks like. Each of these story maps starts with a welcome or ekomomai page, an introduction, and shares wind, cloud, and rain names specific to that island. The remaining pages share geology field guides and mo'olelo, which is a story or legend, that are relevant to those field guide stops and or sites of interest. These story maps are categorized as island-specific story maps. The sixth story map pertains to the state of Hawaii. Unlike other story maps, it's a topic or theme-oriented, with each page corresponding to a different theme. Below are examples of what a page from those story maps looks like, or this story map looks like. Sample themes or pages include Hawaiian Sciences Culture, Site Cultural Protocol, and weather forecasting. Weather forecasting. Each of these pages highlights the interconnection between Hawaiian cultural values, traditional practices, and geoscience. This story map is categorized as a topic-specific story map. Finally, I shared the Esri story maps with SOEST-affiliated geoscientists and educators and revised them based on formative feedback received. In summary, place-based education provides personal learning opportunities, enriches traditional academic instruction, and emphasizes the interconnection of system relationships. Utilizing PBE and geoscience can inspire students to engage in content, pursue relevant careers. The goal of my research is to promote and expand PBE within SOEST by assisting faculty to introduce more and higher level PBE practices into their classrooms. Ultimately, this will help SOEST contribute to preparing a local, diverse geoscience workforce that can help meet the current and future workforce needs of the state of Hawaii. Mahalo anui loa. Thank you very much for your time. Questions, anyone?
Thank you, Tineill. Applause from the audience for your talk. And we do have a question from the audience. So if you can bring your slides down, we'll take this question from the audience as we bring up our third and final speaker for the evening. So Gina, you can go ahead and start your screen share and we'll take this question. It's from Robert Slanau. Would you like to read your question out loud?
Sure. The question dealt with culture, and I realize that Hawaiian culture is the key culture, but our other cultures integrated, and what happens in culture sometimes is very different in different time frames. So I was curious if there was any focus on a specific time frame. So basically, it was questions about the word culture, what it might mean, and all of its ramifications.
Ooh, that's a really good question. In my research, I focused on culture in the sense of traditional practices, land use practices, observations from Hawaiian that's transferred into the Hawaiian language, into hula. So and the focus was not just culture in a broad sense, but was any cultural, more traditional knowledge related to geoscience within the islands. Culture obviously is defined very broadly, but I'm thinking, I'm viewing it through a perspective of traditional Hawaiian knowledge in regards to geoscience, and not just, not everything about culture. I'm just trying to focus it more on as it relates to geoscience.
Okay, thank you.
Mahalo, Tineill. Okay, let's move to our third and final speaker for the evening. Gina, whenever you're ready, you can take it away.
Thank you, Maya. Aloha nui, everyone. So again, my name is Gina McGuire. I'm excited to be the last talk of the day. Thank you all so much for coming and being here tonight. So tonight I'm going to be sharing the work that I've been a part of with Dr. Leah Bremer. I'm sorry. So sorry, Leah. She's here in the audience, to understand the benefits and potential for agroforest restoration across the Hawaiian archipelago. But before I get into what agroforestry means and what our work is, I'd like to introduce myself a little, and also to thank the UHC grant and also PICASC for supporting this work. So meka ha'aha'a, I'm very humbled to be here tonight. A little bit about me. I grew up on Mountain View on Hawai'i Island on a native plant conservation farm, and so kalamai, but I know this is Hanauma Bay, so blessed that you guys have these talks, but most of my examples are from Big Island, so you'll just have to roll with that. But I grew up, my dad grows maile on our farm, as he was taught by the old men's from down Kalapana side. And so I grew up on a farm where my dad was cultivating an ohia and hapu'u canopy to support his lay practice. And so what I didn't realize at that age about learning about things like soil health, about taking care of the stream on our property, and how much work restoration and working on a farm really takes, that would be the roots and the foundation for what I'm now thinking about professionally. So I went on to get my bachelor's in international relations from Stanford, where I started thinking about environmental policy, and I worked with the Nunako Nation on Minjeraba or Stradbroke Island, thinking about Indigenous Australian approaches to marine mammal management. And then I returned home, and I'm so fortunate to have gotten my master's in tropical conservation biology and environmental science at UH Hilo, where I did work on Midway Atoll, thinking about landscape ecology and their visitor program. And I'm now super stoked to be working on my PhD, thinking about place-based well-being from oceanic indigenous perspectives in coastal areas. So I bring all of this love with me to my work, and with this super fantastic team, and I'm sorry their pictures are a tiny bit blurry, but I just wanted to highlight some of the amazing people that I work with. So I said Dr. Leah Bremer, Dr. Tamara Tickton, Dr. Kupika Winter, Dr. Jay Delaveau, Natalie Kurashima, and then the other grad students, so Matt Ko'ohanohano and Zoe Hastings, just we're actively working to understand where and how diverse agricultural systems can enhance well-being through benefits to soil health, food production, and nearshore ecosystems. And so we've gotten back to agroforests, but so what is an agroforest? So an agroforest refers to any diversified agricultural system, and it covers a wide continuum of different land options and systems. Anything that integrates both trees and crops, including animals. We call that silvopasture. So it can be anything from a few trees in a field or a pasture to a multi-storied forest that's stewarded. And we focus on these reciprocal benefits that we get in these systems. And so use of these systems can range from reducing societal impacts. On the far left, you can see intercropping of annuals, to remediation, rehabilitation, and then there's different levels of ecological restoration. So there's different approaches. So for example, Dr. Becky Ostertag at UH Hilo, her work Liko na Pilina is a really great example of a system that mixes both native and exotic species to restore functional forests. So rather than trying to restore a fully native forest, we've got things like mango and kukui as canopy trees, as well as natives such as ohia and things like mamaki. So full recovery would refer to a primarily native ecosystem that's culturally stewarded and gathered from, and that's on the far right of this continuum. So I thought I would go through a few examples. So there's several ranches around the state that do open koa forested cattle ranching. And so these photos are from Kealia Ranch on in South Kona on Big Island. But there's also a active restoration project for koa pasture land at Haleakala Ranch on Maui. And other sites embody that functional restoration approach that is similar to what Dr. Ostertag looks at, where soil retention and functional traits are rehabilitated with exotic species. And this spot is really special to me. So this noni plant is from my dissertation area. It represents an area of historic traditional agroforestry systems that are now predominantly native. So we see these shifts. It's not only kind of, anthropogenically, we might set something up, but then, after we create an agroforest system, what happens after we stop taking care of it is something else. So our work is informed and grounded in an active restoration project at Pu'ulani within the land managed by the nonprofit Kako'oiwi in Heia, Oahu. And so these photos show the process of clearing the result after we I would take no credit, I wasn't there, but amazing team. Our team cleared out the invasive species from this area and planted native and agroforest species. And so you can see iholena in the background. We also have pohinahina, aveoveo, lolu, among other things. And so I'm just really proud of all of the work that everybody has put into that place. But so the question is, why is agroforestry important, especially here in Hawaii? And so in Hawaii, the decline of traditional and plantation agriculture has led to large areas of fallow land in close proximity to the coastlines that provide little in the way of ecosystem services and also pose management risks as far as harboring invasive species and increased fire risk. So the Hawaii Department of Agriculture reported that in 1980, there were over 350,000 acres in crop production, 85% of which were sugar and pineapple. But then in 2015, that was the number of acres in crop production was less than half, and sugar and pineapple had declined to 28% of that area. And diversified crops only accounted for 16,900 acres. And that diversified crops includes stuff like root and melon crops, which are mainly consumed locally. But Loken Leong reported in 2013 that 87% of the food consumed here is imported. So there's a definite need to understand our landscape of agriculture, pasture, and forest. And this will allow us to plan for the potential benefits of agroforestry to maximize ecosystem health alongside economic and subsistence benefits. So for our project, it's nested at 3 different scales, including the site scale that I talked about at Pu'ulani, where we're managing or measuring management costs and challenges, ecosystem services, biodiversity, and biocultural outcomes. And so there's 10 different plots with different treatments of culturally and economic important species to maximize functional traits within stages of succession. And so this is primarily the work of Zoe Hastings and Dr. Tamara Tickton. And soil health metrics are also being taken, percent carbon and microbiology activity, and long-term monitoring for erosion control. Our second scale of work is at the Heia Ahupua'a level and state scales, developing diverse agricultural scenarios for restoration. So at this scale, we're considering a mosaic of agroforests that respond to different goals. My lights are flickering, I'm sorry. To different goals, including sediment retention, economic returns, timber and non-timber products of cultural value. This mosaic of scenarios is supported by interviews with local producers and farmers who are already practicing agroforestry. And so if any of you identify as a producer or grower, please feel free to check out this website, Ho'olu'ulu. You don't need the okinas in there, just ho'olu'ulu.org for more information. And last but definitely not least, we have the scale that I'm most focused at, which brings all of that information from the first two scales together. And so this is developing statewide scenarios. So to maximize all of those ecosystem services I mentioned earlier, so soil health, soil retention, biodiversity, food production, cultural value, functional traits, and economic value together. So in our toolkit to understand connections between land cover types and nearshore ecosystems, we have open source models available from Stanford Natural Capital Project, and they have a variety of models such as habitat quality and coastal blue carbon, a whole bunch. But to understand the potential for agroforestry restoration, we're using the sediment delivery ratio model. So this model considers climate, mainly rainfall intensity, soil properties, topography, vegetation types, and then human activities and how these influence overland erosion. And in Hawaii, our nearshore ecosystems are directly linked to sediment delivery up Mauka. And the USGS, they documented in 2007 that sediment runoff is the biggest land-based pollution threat to our reefs. And so sediment runoff, I'm sure you guys are all familiar with this, but it causes deposition and accumulation, resuspension and high turbidity, reduced coral reproduction and recruitment, and just generally less coral cover. So to another ecosystem service we're modeling is carbon storage potential of these diversified ecosystems. And so in 2016, Stefano and Jacobson documented, they did a literature review, and their findings were that there were significant soil organic carbon stocks that increased when you convert land from conventional agriculture, pasture, and grassland and uncultivated lands to these agroforest ecosystems. And their findings looked at work that was done across North, South, and Central America, Africa, and Asia. And so alongside sediment delivery, we'll also be modeling potentials for agroforestry restoration here in Hawaii, and so the benefit to our carbon sequestration. And so to model these ecosystem services, most of my work so far has been prepping the map model inputs. It's very, some of it is time consuming, but these layers include rainfall erosivity, soil erodibility. So rainfall erosivity is basically just how hard the rain is hitting the ground, and that's based on elevation, it's based on vegetation cover, and then that's related to soil erodibility, which is, you know, how type of soil and how easily it washes off of that particular cell grid that we're looking at the state level. And then land cover type, a digital elevation model, and a watershed layer. And so I want you guys to kind of focus and look at the two in the center. So at the top, we have the 2015 Carbon Assessment Hawaii land cover map. And then on the bottom, we have the 2015 agricultural baseline. And so for our land cover map, what we really wanted to do is understand not only the vegetation cover, which the carbon assessment map gives us in really high detail. So you can tell what kind of forest it is, you can tell if it's invasive grassland or native grassland, but we also wanted to understand how it was being used. And so the ag baseline was made using data from aerial imagery, but it was also combined with state and county data sets, so tax map information and water use data. And so when we put these two maps together, the carbon assessment one allows us to understand the array of vegetation on a parcel. For example, the fringe of Ohia forest that borders a papaya farm, while the ag baseline would label this whole area as papaya. But on the flip side, the ag baseline would allow us to understand land cover based on use. So while the carbon assessment layer would allow us to see, okay, this is an open acacia koa forest, but when you combine it with the ag baseline, we learn that it's fenced within a pasture. So we have unique values where the ag baseline identifies land use for aquaculture, banana, coffee, commercial forestry, dairy, diversified crops, flowers, foliage, and I'm like rushing through this list, but macadamia nuts, papaya, all those kind of different things. And then those were substituted into the land cover map where it generally just marks the land as ag land. And then pasture was overlaid so that we can understand the composition of our pastures here in Hawaii. So from this layer, what I'm currently working on is assigning each different land cover type a C-factor value, which is basically just the effectiveness of soil and crop management to preventing soil loss. So this is based on previous studies and an intensive literature review to develop ranked values. So for example, a full native forest that's fenced without ungulates, really high quality forest, might have a higher C-factor value than ungulate disturbed invasive grassland. And Hawaii is a unique area to develop these C-factor values with our really unique volcanic geology. So our next steps are to identify areas that might benefit from agroforestry restoration at the state scale and provide a mosaic of restoration scenarios that might benefit these areas and communities. These scenarios are a combination of the information we're getting from our practitioner interviews, identifying what grows, where and why, as well as contextualized within different goals for subsistence and community sustenance, local food production, and potential export items. We also acknowledge that agroforestry is not something new here in Hawaii. And so super stoked to be working with Natalie Kurashima, and she developed these spatial layers that identify the potential for indigenous agricultural food production. And her research shows that urban development has reduced that traditional agricultural areas by 13%, but 71% remain agriculturally zoned. So there's a lot of opportunity there. And indigenous agricultural systems have demonstrated resilience, diversity in species assemblages, and to provide potential overlaps in native species biodiversity. And it's based in kinship ties to food systems, which are often overlooked in academia and policy. So we hope to highlight both native and canoe plant agroforests as key part of these scenarios to sustain that connection both to our kupuna and the kupuna of the Aina, as well as to our piko of who we are now. For our communities today to be healthier, more independent, and reciprocally benefiting from these Aina-based relationships, but also to create, a vision for an economy and a landscape that our future generations can be proud of. And again, I just wanted to acknowledge my team because they're awesome and I love working with them, and also to say thank you so much to Sea Grant and PICASC for supporting us and all that we do. Specifically, I'd like to say thank you to Leah and Jade and Tamara for supporting me. And so we hope that this archipelago-wide identification of hotspots, as well as agroforest-specific scenarios, will highlight potential opportunities for the state to restore its historic high-intensity agricultural lands, to envision a future that is more self-sustaining and more conscious and beneficial, including to our reefs. And with that, I think I'll open it up to questions. And thank you all so much for listening. I really appreciate it.
Mahalo, Gina. Applause for your talk. Thank you so much for putting a great talk together tonight. Lots of questions actually in the chat.
I know, I'm kind of scared.
We are going to end on time. We're going to end at 7:30. Maybe this question from Julian. Julian, would you like to read your question?
Sure, I'll read it. So I was wondering if there's any downfalls to growing functional forests as opposed to all natural forests.
Yeah, thank you so much, Julian, for that question. I think, I think ideally we would all love to have native forests and endemic species and to really restore ecosystems to what was here pre-contact, or even including those canoe species. But I think the benefit of these functional forests is that native species sometimes don't compete as well with invasive species as these other canoe plants might, or other species can, and they're also, some of them are quicker growing, so there are downfalls, not being able to restore a completely native food system, or others outside of food, medicine, and timber, and but I think doing what you can, and there's nothing wrong with being a little innovative. But yeah, that's a great question. Thank you so much.
Okay, we welcome questions for any of our speakers. So as a reminder, we had a great talk about larval fish and their prey items tonight from Cassie and Emily. An awesome talk on place-based education and the geosciences from Tineill, and then Gina, who provided a great talk tonight on agroforestry and some of the research that she's engaged in. So I'll let those questions trickle in. Let's take another one. Emily, do you want to ask yours?
Sure. This is a little bit of a technical question, but what software do you normally use for your spatial analyses?
It's primarily in ArcGIS right now. I did a little bit of the preparing the watersheds and like the hydrology stuff in QGIS, which is the open source platform version. But yeah, primarily Arc, and then a little bit of it is done in R, so coding. But yeah, thanks, Emily. It's a great question.
Thank you.
Okay, let's see. I see many different questions from, oh, and here's a new one. Ooh, this is a great question from Barb. Barb, would you like to read your question? I think this is how we'll end the evening.
Sure. As this is my first series of talks that I'm attending as part of the Hanauma Bay Education Series, I was curious, and this is a question to all of the presenters, of what aspects of the science communication training that you received through the Sea Grant and PICASC fellowships was most helpful to you in preparing this talk. So I don't know if you want to perhaps answer this in the order you spoke, or whatever you think.
Right, that's a really good question. So this is kind of a simple answer maybe, but during our training we talked a lot about jargon, and it's still definitely a learning process for me, but as scientists we sometimes don't notice how much jargon we're inserting into our conversations, like super technical words. So just stepping back and making sure everything is understandable, I think, is super useful.
I would have to second that as well. And just general science communication for me in graduate school was I was able, as a graduate student, to present my my presentation to a number of people. And so when I got the feedback, I learned what I was making clear and what was not so clear to my audience. And I really think that helped, because as I'm making, you know, the slides, it makes sense to me, everything's clear to me, but my audience, it might not come across that same way.
Same for me, jargony, and I think that was still evident a little bit in my talk. but I practiced the recommendation that I took from the training was, you know, to practice on people who might not have a science background. And so my parents, who are farmers, so they understood a lot of what I was talking about agroforestry and farming, but I ran it past them, and they told me, you know, make sure if you're talking numbers like I did with the current state of agriculture, make sure that's on the slide, because people can't, you know, small stuff like that. But thank you guys so much for the training that you provided. Really appreciate it.
Mahalo, Gina, Tineill, and Emily, for those awesome answers. We're going to end it here. Thank you so much for joining us tonight. We are hearing from our Sea Grant GRA fellows next week, postgraduate fellows. So come back next week if you're interested to hear about their work. Thanks, everyone, for coming.
Thank you for joining us. I hope you guys can join us for next week.